So let's talk about regulating gene expression. There's several syllabus objectives here, so I want to put these up again at the end so you can go back and reflect on whether we've covered off on all of these syllabus objectives. And I'm also going to put the syllabus guidance up as well so that you can look at what you don't need to know. Okay, so let's get into it. So as we've said in the previous video, gene expression is the process by which information from a gene, the sequence of the nucleotides in a gene, is used to synthesize a functional gene product. So of course, functional gene products can be structural proteins, like what makes up our muscles and our bones and our skin. Also, don't forget functional proteins, and they include enzymes as a classic example. But another one is gene expression regulators. So this video is all about regulating gene expression. So we have genes that are actually involved in regulating gene expression. And so some of the products of those are proteins, functional proteins, that actually regulate the expression of other genes. We also, of course, have functional gene products being functional RNA like mRNA and tRNA. So a really important point here is, as you know, each one of the cells in any organism contains the whole genome, every single gene. Many of the genes won't be expressed in a particular cell because the cells are differentiated to do a particular role, whether it's to make tears or make insulin or make hydrochloric acid. And you don't want the wrong cells doing these things. So most genes are never actually expressed in a particular cell. And even if they are, they're not expressed all of the time. The process of regulating gene expression is controlling what genes are expressed and when they're expressed. Put simply, gene regulation regulates the phenotypic expression of genes. And there's a number of different ways in which gene regulation occurs. It's very complex. We're obviously not going to deal with it in a tremendously complex way. The first way we're going to look at it is through the process of transcription and translation. So before and after transcription, and before and after translation. Pre-transcriptional regulation is related to transcription factors. It's basically whether the transcription process starts in the first place. So we have these transcription factors that are RNA or proteins that are the products of regulatory genes, and they actually need to bind to the start of the gene to activate the transcription, or equally, they might repress transcription. These are called transcription factors. We're going to look at them in more detail. So, for example, we have transcription factors that activate. So they actually bind to a specific area of the gene, the start of the gene. And what they do is they actually help the RNA polymerase assemble, ready for transcription to occur. But equally, we have transcription factors that are repressors, and when they bind to the site, then no transcription occurs. After transcription, there's another opportunity for regulation. And so one of the things that happens is that the introns, that non-coding part is spliced out. So it's snipped out and joined back together. But also there's an opportunity for alternative splicing in some cases. So the same mRNA copy of the gene can be modified to end up producing a different protein. And a great example of that is for the difference between skeletal muscle and smooth muscle. They're both types of muscle, but obviously they're quite different structurally, but the same gene codes for them, and we actually have post-transcription modification. There's also translational regulation as well. So we might have mRNA, but it might actually block the start of translation. And then also post-translation, this is when there's modifications to fold the protein into its functional or structural final state. So I mentioned that some genes are regulatory genes. So they produce proteins that regulate the expression of other genes, and they're called transcription factors. One good example of that is what's called the lac operon. So this occurs in prokaryote cells, So an operon is where you've got a number of genes that get transcribed into one messenger RNA, so that then they, through the process of protein synthesis, the gene products can be produced at the same time. Now, operons are really common in prokaryotes, but not in eukaryotes. And the lac operon, LAC, lac, that's short for lactose. So this process is involved in the digestion of lactose in prokaryotes, and it's a classic example that is talked about. So we'll briefly talk about it here. Okay, so this yellow thing here, this is RNA polymerase. So as you can see, when there's no, this stuff here is lactose. So when there's no lactose around, we have this repressor that's bound to the start of the gene, and it prevents RNA polymerase from being able to start the process of transcription. Now, when lactose is present, it binds to this repressor and removes it, allowing the RNA polymerase to be able to bind and start the process of transcription. So this is a classic example of a transcription factor. It's actually the environmental conditions that influence whether the transcription is allowed to occur or not. So we have regulatory genes that produce a functional gene product that controls the transcription or the expression of another gene. So the example of the lac operon is an example of where there's environmental exposure that influences the expression of the genes. And another one is the insulin gene, that when glucose levels are high, more insulin is produced. So we can use gene regulation to control homeostatic response to changes in the environment. So another way in which gene expression can be regulated is through epigenetic control. So epigenetic means near or above the genome. So we have a number of internal and external environmental factors that can actually influence the expression of a gene without changing the nucleotide order. They add chemical tags to the DNA, and it influences which genes are expressed and how much they're expressed without actually changing the genome. So there's a whole range of different epigenetic factors relate to diet, time of year, disease, toxins like chemicals and drugs and alcohol and things, your financial status. Other things include exercise, your microbiome and your gut, socializing, your psychological state, alternative medicine, and obviously therapeutic drug. So this is a great opportunity in terms of the future of therapeutics, in terms of how diseases can be managed. One way in which the epigenetics has been studied is through the twin methodology in epigenetic studies. So we can get two identical twins that have had different environmental exposures and looking at any differences in gene expression. So we've established that every cell has the genome in it, and this is important for the development and differentiation of a complex organism, multicellular organism like a human. So some genes are expressed at the fetal stage or the embryonic stage and don't need to be expressed later in life. And one example of that is called the Hox genes. They're expressed very early in embryonic development, and they're involved in determining the anatomical structure of the organism. So they're regulatory genes in terms of producing functional gene products that are transcription factors, and they're involved in determining where appendages are attached to each of the body segments. So there's a number of genes, and they determine where the arms grow and where the legs grow, and where the organs are in our body. So they learned about these genes by manipulating the genes on these flies, and for example, they were able to manipulate them such that another set of wings was grown, or that a leg grew where an antenna should be. This is in the Drosophila fly. But also, here's some examples of gene disorders of the human Hox gene. So another gene that's really important is called the sex-determining region Y, or the SRY. Essentially, all human embryos have the capacity to either develop into male or female reproductive tract. And the default is for it to develop into female characteristics. So there's a gene on the Y chromosome, so only males have this gene and only males express this gene. And it produces a transcription factor, which is a protein that binds to the DNA and it regulates expression of other genes. Specifically, what it does is it stimulates the formation of the testes really early on in the embryonic development. And it's the testes that produce testosterone. And that inhibits the female hormones and overrides the formation of the female reproductive structures such that the male reproductive structures develop. In the absence of the SRI, genitals continue in the female development. Okay, so this video is all about regulating gene expression, and there's a number of syllabus objectives, so take a few minutes to actually read through the syllabus objectives to identify what you specifically need to know. And also have a look at the syllabus guidance to look at what you don't need to know. When identifying transcription factors in the regulation of gene expression, you don't need to get bogged down in all of this detail about all of these things. This is more than you need. You need to be able to explain gene expression in the context of an example. So we talked about the lac operon, we looked at the Hox gene and the SRY as well. But you don't need to be able to explain or describe those mechanisms, just have an understanding of the examples.